Good morning. I hope you're all doing well.
Good morning.
I'm good. Thank you, man. Let's see. All right. Um, Still waiting on two. All right, let's get started. And we are going to start the second unit today on coenzymes and vitamins. So this is chapter 7. Let me just sharing my screen with you all. All right. So our exam this Friday will cover Unit 1 from chapters 1 through 6. Okay? And then for Unit 2, which will start with chapter 7, covering all through chapter 11, this will be included in your exam too. But just like what I've mentioned on the first day of class, when we have time, we start another chapter if there's no problems to work on. And hopefully by doing this, we will be on track with our schedule. And if we, excuse me, if we get ahead of schedule, that will help. Um, just a heads up, there may be a couple of days in July. I don't know the exact days yet that I may be going out of town. So by being ahead of our schedule, that will help when we don't meet those days. All right. Okay. So for chapter 7 on coenzymes and vitamins. Cofactors may be divided into two groups, those that are essential ions, of which most of them are metal ions, and those that are coenzymes that consist of organic compounds. Right? And within each group or groups of these cofactors, they are subdivided further. In both cases, their subdivision is based on how tightly the groups are bound to the enzymes. Loosely bound essential ions are termed activator ions, while um, tightly bound ions are found in metalloenzymes, right? Loosely bound coenzymes are classified as co-substrates, while tightly bound coenzymes are designated as prosthetic groups. So those are the subdivisions within these two main groups of cofactors. Now, over 25% of all known enzymes require metallic cations to achieve full catalytic activity. Metal activated enzymes, they either have an absolute requirement or they can just be stimulated by metal ions. These metal ions include potassium, calcium, and or magnesium. More firmly bound metal ions and in metalloenzymes include primarily iron, zinc, with copper and cobalt being encountered less often. Right. And let's just look at one example on how these are involved in catalytic activity. So our example will be iron. This is a metal ion that's commonly encountered in metalloenzymes. It participates through reversible oxidation reduction reactions. And some enzymes, they have heme groups. Is it? All right. So as I've mentioned, some enzymes, they have heme groups, and those heme groups contain iron. Cytochromes are also contained in enzymes, and they contain also iron. We'll be more familiar with this, especially when we, for the cytochromes, when we cover chapter 14, right? But these heme groups, again, they have iron, and cytochromes also contain iron. There are also non-heme iron that exists. So iron is not only associated with heme proteins and enzymes, but non-heme iron exists in what we call iron sulfur clusters, just like what we see here in these figures. So in these iron sulfur clusters, the sulfide ions, as well as the thiolate group, remember cysteine, it has the thiol group. This will bind the iron. Okay, the sulfur or the sulfide and the thiolate groups will bind um, the iron. Iron sulfur clusters, they accept only one electron at a time. Right? And whether there are two or four, as we see here on the right, four iron ions associated with the complex, again, the complex can only accept one electron in an oxidation reaction. Right? So for this iron, iron undergoes reversible oxidation and reduction. Another group of cofactors are coenzymes, and coenzymes act as group transfer reagents. Um, when we say that coenzymes act as group transfer reagents, the coenzyme can transfer hydrogen, electrons, and other groups, okay? Those groups that can be transferred. There are two classes of coenzymes. Co-substrates, they are altered during the reaction. They are loosely bound and regenerated by another enzyme. When the coenzyme remains either covalently or tightly bound to the enzyme during the reaction, then it is known as a prosthetic group, right? So for these two classes, please remember, co-substrates loosely bound, right? They're changed during the reaction and then regenerated by another enzyme. While prosthetic groups, they are tightly bound to the enzyme. A further means to classify coenzymes is based on their source. coenzyme that mammals can synthesize from common metabolites are referred to as metabolite coenzymes, while coenzymes that are based on vitamins that mammals need in their diet, there are the vitamin-derived coenzymes. So for the rest of this chapter, we will go over examples of each type of these coenzymes. An example of a metabolite coenzyme is the nucleoside triphosphate, adenosine triphosphate. There are actually a number of nucleoside triphosphates that serve as metabolite coenzyme, but by far the most abundant of those is ATP. ATP consists of a nitrogenous base, so this adenine base here, linked to a ribose bearing 3 phosphate groups. So ATP is a versatile reactant that can donate phosphoryl group. This is the most common group donated. So when ATP is cleaved and hydrolyzed into ADP and inorganic phosphate, right? So this inorganic phosphate here can be donated to another molecule, and this inorganic phosphate is the gamma phosphate, this part here, this phosphoryl group here. Pyrophosphoryl group can also be donated, and the pyrophosphoryl group consists of the beta gamma phosphates. So this is the pyrophosphoryl group here, and that is a product of the hydrolysis of ATP into adenosine monophosphate, and there's the inorganic pyrophosphate. The adenyl group, or AMP, which is highlighted in blue in this figure here, that's the next most common group that is donated. So the most common group donated by ATP is phosphoryl group. The next common group that is donated is AMP. The adenosyl group, which consists of adenine and ribose, can also be donated. So ATP, again, is a versatile reactant that can donate these different groups in reactions. Another metabolite coenzyme is adenosyl methionine, or SAM. And this compound is formed by the reaction of methionine with ATP. We see here, so methionine reacting with ATP will produce SAM. And the activated methyl group here is shown in red. The positively charged sulfonium here of S-adenosyl methionine is highly reactive. Now, what is the role of S-adenosyl methionine in biosynthetic reactions? SAM donates methyl groups in many biosynthetic reactions. Actually, S-adenosyl methionine is the donor of virtually all the methyl groups in biosynthetic reactions. The general reaction showing methylation of compound X is shown here. So the methyl group from S-adenosyl methionine is donated to compound X. So we now see this methylated compound X here. That is the general reaction. Nucleotide sugars like UDP glucose are also metabolite coenzymes. UDP glucose is formed in the reaction of glucose 1-phosphate with uridine triphosphate. And UDP glucose that is formed from that reaction can donate or transfer its glycosyl group, which is highlighted here in red. So this glycosyl group can be transferred to a suitable acceptor, and in the process releases uridine diphosphate. Nucleotide sugars, again, the role transfers sugars in carbohydrate metabolism. And the specific example that we looked at here is UDP glucose, which transfers the glycosyl group to a suitable acceptor molecule. Another metabolite coenzyme is lipoamide. And the coenzyme lipoamide is a protein-bound form of lipoic acid. The lipoic acid is bound to the epsilon amino group of lysine, as we can see in this structure down here. The reactive center of the coenzyme is shown in red. Right? So this here is the reactive center. So to remember the structure of this lipoic acid, remember that this it has two sulfurs of the carbons 6 and 8 in this in a disulfide linkage that is shown here, and that again is the reactive group. The role of lipoamide is that it acts as a swinging arm that carries acyl groups between active sites in multi-enzyme complexes. Right? We'll see this when we cover, let's see, chapter 13. Okay. And going into chapter 14. Now the acetyl group, say for example, instead of this sulfur here, this is replaced by an acetyl group, right? This sulfur here. The acetyl group that is attached to carbon number 8 of lipoamide, it can be transferred to acceptor molecules. So role of lipoamide again is that it acts as a swinging arm that carries acyl groups between active sites of enzymes. Ubiquinone, that is also known as coenzyme Q. It is a lipid soluble coenzyme. Right? It is synthesized by respiring organisms, and some photosynthetic bacteria also synthesize ubiquinone. The role of ubiquinone is to transport electrons between membrane embedded complexes. You'll be very familiar with this as an electron mobile carrier in the electron transport chain when we cover chapter 14. So the role again is to transport electrons between membrane embedded complexes. Hence, being a lipid soluble coenzyme, that's very important. the hydrophobic tail. So what can help us recognize the structure of this coenzyme? Look at the structure in figure A. It has this hydrophobic tail here, and it is composed of 6 to 10 isoprenoid units. Right? The hydrophobic tail again is composed of 6 to 10 5-carbon isoprenoid units. This figure B here is plastoquinone. It is an analog of ubiquinone, and it functions in photosynthetic electron transport. Okay. Ubiquinone is a strong oxidizing agent. It can exist in 3 oxidation states. The fully reduced state is ubiquinol. The partially reduced state is the semiquinone free radical. And the oxidized state, the fully oxidized state, is ubiquinone. So we can see here the 3 oxidation states of ubiquinone. Because ubiquinone can exist in 3 oxidation states, this coenzyme is capable of accepting or donating electrons 1 or 2 at a time. So ubiquinone again can accept or donate electrons 1 or 2 at a time. Right, to sum up what we've just covered on the examples of metabolite coenzymes, we examined ATP, S-adenosylmethionine, or SAM, UDP glucose. We also examined lipoamide and ubiquinone, and I mentioned about heme group. So ubiquinone, lipoamide is prosthetic group, ubiquinone, and the rest here, ATP, SAM, uridine diphosphate glucose or UDP glucose, they are co-substrates. Right? Ubiquinone is also a co-substrate. Now we shift into vitamin-derived coenzymes. These are coenzymes whose precursor must be obtained from the diet, and then they are enzymatically transformed to the coenzyme. Animals rely on plants and microorganisms for vitamin sources. Let's start with NAD or NADP. Nicotinic acid is the precursor of NAD and NADP. Both the coenzymes NAD and NADP contain a phosphoanhydride linkage. So we see the linkage here and for the reduced form here. Right? So NAD, NADP contain a phosphoanhydride linkage that joins AMP and NMN, nicotinamide mononucleotide, and AMP, adenosine monophosphate. The difference between these two coenzymes, NAD and NADP, is that NADP has a phosphoryl group at the 2' oxygen of the adenylate group here. All right. So say, for example, we are looking at the structure and it just says OH here at the 2'. Without the phosphate group, that will be NAD. But if we see that the 2' is phosphorylated, then that is NADP. On the right are the reduced forms. How do we distinguish the reduced forms from the oxidized form? Look at the hydride ion attached to carbon number 4. So here, right, that carbon number 4 there is a hydride ion attached to it. So whether it's NAD or NADP, if a hydride ion is attached to carbon number 4, then it is the reduced form. NAD and NADP serve as cofactors in oxidation reduction reaction. So this is the role of NAD and NADP. They act as substrates for dehydrogenase enzymes. Now, in the mitochondria, NADH, the reduced form of NAD, is coupled with ATP production, while NADPH is an important reducing agent in a lot of biosynthetic reactions. One of the ways to monitor enzyme activity requiring these cofactors, NAD and NADP, is to use a spectrophotometer at 340 nanometers. So this reduced forms of NAD and NADP, they absorb UV light at 340 nanometers. Right? The oxidized forms, NAD and NADP, do not. So that is one of the ways to monitor enzyme activity requiring these cofactors, to monitor using, say, a spectrophotometer at 340 nanometers, the levels that is present in the reduced form. Because again, no absorption of light at 340 nanometers by the oxidized form. One example of an enzyme requiring the NAD coenzyme is that of lactate dehydrogenase. This lactate dehydrogenase enzyme catalyzes the reversible, because we have 2 arrows here, so we have a reversible reaction, the reversible oxidation of lactate into pyruvate. In conjunction with the reduction, with the oxidation of lactate to pyruvate, there is reduction of NAD to NADH. Okay. So a proton is released from lactate, and that is used to reduce NAD to NADH. One of the distinguishing features of lactate dehydrogenase enzyme, which is actually typical of dehydrogenases, is that it follows a sequential ordered mechanism. Remember last time we looked at sequential and ping pong, right? This follows a sequential ordered mechanism wherein the coenzyme is first bound to the apoenzyme to form a holoenzyme. Okay. The holoenzyme then binds the substrate, and in this case, it's lactate. Reactions will occur, and then products will be released, pyruvate, NADH, and the enzyme is regenerated. So again, we see here the sequential ordered mechanism of reaction. Another vitamin precursor is riboflavin, vitamin B2. Flavin, adenine, dinucleotide, and flavin mononucleotide are derived from riboflavin. Right? For this structure, riboflavin actually consists of this 5-carbon ribitol here, right, which is linked to a nitrogen atom of this isoalloxacin. So this whole thing here is the vitamin precursor riboflavin. FMN then, flavin mononucleotide, is a phosphorylated riboflavin. The structure that is highlighted in black, that is flavin mononucleotide. So it consists of the precursor vitamin B2, which is then phosphorylated. How about FAD, flavin adenine dinucleotide? It contains this AMP and FMN. So the FMN, when it is bonded to AMP, this whole structure now is flavin adenine dinucleotide. What is the role of flavin coenzymes? Flavin coenzymes are involved in oxidation reduction reactions. When we compare FAD and FMN with, say, NAD and NADP, remember that NAD and NADP, they are capable of conducting the 2 electron transfers, I should say. So NAD and NADP capable of conducting 2 electron transfers. FAD and FMN catalyze either 1 or 2 electron transfers. So here in the quinone form, 2 electron so 2 protons. Remember that there's kinetic coupling between electron and proton transfer, right? So quinone form fully reduce into this hydroquinone form. Okay. And then with the loss of 1 proton, a semiquinone form is generated. Further loss of this proton here will then produce the quinone form. These coenzymes, FAD and FMN, they can also be easily oxidized in the presence of air. Hence, to prevent oxidation in the presence of air, the coenzymes FAD and FMN are usually tightly bound to their respective enzymes to prevent exposure to oxygen. Coenzyme A is derived from vitamin B3. Metabolic processes that require coenzyme A include breakdown of carbohydrates, fatty acids, amino acids. So the breakdown of those macromolecules involve coenzyme. It involves coenzyme A. Also by synthesis of some carbohydrates and lipids involve coenzyme A. CoA has 3 major components. Okay? Based on the structure, there's the mercaptoethylamine, and then a pantothenic acid, and then this ADP with a 3' phosphate group. Right? And the reactive center is this sulfhydryl group here. The role of coenzyme A is that it participates in acyl group transfer reactions with carboxylic acids and fatty acids. So that is the main role of coenzyme A. It is a donor of acyl group. It participates in acyl group transfer reactions. So we say that SAM transfers methyl groups, coenzyme A transfers acyl group, right? So where are the acyl groups? We're looking at the coenzyme A structure here. Where will be the acyl groups attached to? The acyl group will be attached to this reactive center here, the sulfhydryl group. That will then form a thioester. There's a variation of coenzyme A. It's called the acyl carrier protein. And the acyl carrier protein is esterified to a serine residue of a protein. To distinguish then the structure between coenzyme A and acyl carrier protein, for coenzyme A, the CoA is attached to ADP here. But for acyl carrier protein, it is attached to a serine residue, right? Both of them, how do we compare those coenzymes? Both have the sulfhydryl reactive group. All right. Thymine pyrophosphate is derived from vitamin B1. So vitamin B1 serves as the precursor to TPP, and thiamine or vitamin B1 is this group here. Whoops. All right. It contains this possibly charged thiazolium ring. It also contains this pyrimidine ring here. So this thiazolium, this possibly charged thiazolium ring here, serves as the reactive center. How is TPP synthesized? TPP is synthesized by transferring a pyrophosphate to thiamine. So we get this whole structure here, which is TPP. This pyrophosphate here, that is donated by ATP. So pyrophosphate group from ATP transferred to thiamine to produce the coenzyme thiamine pyrophosphate. Right? Remember the gamma beta phosphoryl groups of ATP that are can be donated. This is the pyrophosphate group here. Role of TPP. Well, carboxylases, decarboxylases, those enzymes are known to require TPP as a coenzyme. Right? So this coenzyme then participates in the carboxylation reaction and the transfer of 2 carbon units in carbohydrate metabolism. And then, so I said carboxylases, decarboxylases, those are enzymes that are known to require TPP as a coenzyme. Another group of enzyme for which TPP serves as a coenzyme are the transketolase enzyme. So Transketolase enzyme also require transketolases also require TPP as a coenzyme. When we say transketolase, this is an enzyme that is involved in the transfer between two sugar molecules of carbon groups, okay, that contains keto groups. So transfer of two carbon units, say in carbohydrate metabolism. Right. So we know here the role of TPP, and then next is PLP, pyridoxal phosphate, which is derived from vitamin B6. Vitamin B6 is a phosphorylated form of is phosphorylated, I should say, to form PLP. So this phosphoryl group here, that is donated by ATP. It's the gamma phosphoryl group that is transferred to vitamin B6 to form PLP. Right? When vitamin B6 enters a cell, there's that enzymatic transfer of the gamma phosphoryl group from ATP to form this pyridoxal 5' phosphate. And for its role, PLP is a prosthetic group for enzyme catalyzing reactions involving amino acid metabolism. So PLP is a prosthetic group that is involved in isomerization reaction, decarboxylation reaction, side chain elimination in amino acid metabolism. Okay? And if we refer to this reaction we call transamination, wherein an amino group is transferred, okay, from one molecule to another. The enzyme that catalyzes the transfer of the amino group are called aminotransferase. So picture here that the amino group is accommodated here. So instead of oxygen there, then an amino group is accommodated there. When there is an amino group here, PLP then is converted to PMP, or pyridoxamine 5' phosphate. And that amino group, which will can be found here, that is transferred to, say, alpha keto acids to produce new amino acids. Right? So this is important in amino acid metabolism. Biotin is the precursor, and it is also the coenzyme. Biotin is one of those vitamins that is required in very small amounts because about 1/2 of the recommended daily allowance can be supplied by the bacteria in our gut. Biotin does not require any enzymatic modification for its activity. However, it is covalently linked to the active site of the enzyme by an epsilon amino group of lysine. So this is the epsilon amino group that is linked to biotin. Okay. The reactive center of biotin is shown here. It's highlighted in red. It is at the N1 position. The role of biotin It is a prosthetic group of, I should say, that catalyzes carboxyl group transfer reactions and ATP-dependent carboxylation reactions. So this biotin, it can be an intermediate carrier of mobile carboxyl metabolic group. Hang in there, we have three more. Tetrahydrofolate, THF, is derived from the vitamin folate. And the coenzyme THF has this structure at the bottom where positions 5, 6, 7, and 8 of the pterin ring, of the ring, they are reduced. So this is a good way to distinguish this or to recognize THF, this coenzyme. Also, to recognize this coenzyme, look at the THF containing glutamate. Okay? And it can contain, it says here N, but it can contain 5 to 6 glutamate residues, which will facilitate binding of the coenzyme to enzymes. Role of THF: participates in the transfer of 1 carbon units at the oxidation level of methanol, formaldehyde, and formic acid. So again, tetrahydrofolate is required by enzymes that catalyze biochemical transfers of 1 carbon units at the oxidation levels of methanol, formaldehyde, and formic acid. The largest vitamin is vitamin B12, and it contains this ring structure here. This ring structure is actually similar to the ring structure found in heme. However, in heme, instead of cobalt here, what do we find again? Iron, right? The ring structure in heme contains iron, while this ring of vitamin B12 contains the metal ion cobalt, not iron. So this is the precursor, vitamin B12. What are the coenzymes derived from vitamin B12? Methylcobalamin and adenosylcobalamin. In methylcobalamin, instead of this hydroxyl group here, bound to cobalt, there is a methyl group bound to cobalt, and that coenzyme is called methylcobalamin. Now, how about instead of that hydroxyl group there is an adenosyl group bound to a cobalt. In this case, the coenzyme is adenosylcobalamin, right? So this cobalamin vitamin B12 structure that is the structure is shown here. The coenzymes derived again from this vitamin B12 are methylcobalamin and adenosylcobalamin, and these are the structures for those coenzymes. The coenzymes, methylcobalamin or adenosylcobalamin, participate in molecular rearrangement, in which a hydrogen atom and a second group, designated here as group X, will exchange places, right? And that rearrangement again of this hydrogen atom and group X on adjacent carbons, that involves the cobalamin coenzymes. A specific example is shown here at the bottom: rearrangement of methylmalonyl CoA to form succinyl CoA. And in this molecular rearrangement reaction, adenosylcobalamin participates in the reaction. So this is the role of cobalamin derived coenzymes. They participate again in molecular rearrangements. All right. Okay, so I just included this table here, which provides an overview of the vitamins, and these vitamins are water-soluble vitamins, so they may be readily excreted in the urine, and they are required daily in small amounts. When the amounts are lacking, when there's deficiency, then various diseases are encountered, although these are rare now. So these are the diseases associated with a deficiency of these vitamins here. Yeah, just for a reference. And lastly, I've listed here a list of chapter 7 and of the chapter problems. So after chapter 7, you'll find these problems here. So these are highly recommended. I will not collect the answers to this, but I highly recommend that when you prepare for exam 2, you will be able to study these questions here. So try them first. The answers are actually found at the back of the book, right, of our textbook, okay? Oh, by the way, so we looked at vitamins that are precursors to coenzymes. Remember that there are vitamins that are not precursors to coenzymes. Vitamins like the so-called lipid vitamins, vitamins A, D, E, and K, right? Those are not involved in the synthesis of coenzymes. However, they also have important functions. Like for vitamin A, it can regulate gene expression during cell differentiation. Vitamin D, we know, regulates absorption of calcium and its deposition in bones. Vitamin E and has antioxidant actions and may prevent damage to fatty acids in biological membranes. How about vitamin K? It has been shown to be required for synthesis of proteins in blood coagulation. All right? Okay, so this is chapter 7, and let me share this. The next quiz will be due next week. So that's a Monday. And this quiz covers chapter 7. So you're given different structures, and you're asked to give metabolic function and identify those as these cofactors as co-substrate or prosthetic groups. All right. And there is another question here. So just a heads up, this chapter 7 that we've covered, this is covered in quiz 2, which is due on Monday, June 22nd, no later than 11:59 p.m. Right. Also, another announcement. Your assignment 2 um, is due tomorrow. Tomorrow, yes, the 17th. Okay. About the Michaelis-Menten equation, Lineweaver plot. Right. And to remind the class again, we have on the calendar the due dates of the assignments and quizzes, as well as the dates of the exams here. Okay. For the exam on the 19th. So some of you, you have a class after here. And so instead of starting the exam at 10 o'clock, okay, I will open, the exam will be accessible starting at 9 o'clock, and I will give 30-minute extra time if needed, if you need it, but you'll be given 2 hours to finish the exam. So if you start at 9 o'clock, by 11 o'clock, you should submit your exam. Again, you'll be using Lockdown browser. If you start at a regular time, which is 10 o'clock, then you have to submit your exam at no later than, I should say, 12 o'clock noon. Right? So that's for the schedule of our exam. That way, everyone could get the 30-minute extra time if you need it. And okay, another announcement regarding exam 1. I will prepare a take-home portion of exam 1. And that take-home portion will be worth 10 points. And I will give that take-home portion on Thursday, right? I'll check by tonight. If I'll prepare it, I may open it late tomorrow, but definitely it will be open on, you will access it on Thursday, and then you need to submit it no later than 11:30 a.m. on your exam day. Okay, it will be included, the instructions will be included there. If the take-home portion, if it's submitted after 11:30 a.m. on Friday, it will get a percent deduction. And if it's submitted after 12 p.m. on Friday, it will be a grade of 0, right? So please note the instructions on the take-home portion. Okay, so any questions?
For the Chapter 7 PowerPoint, could you upload that to ELC?
Oh, I'm sorry. Let's see. I think I've already, which one? Let's see. So yeah, Ahmad, the chapters for Unit 2, yes, they've already been uploaded all the way to Chapter 11, all right? As well as the in-class review questions. So tomorrow we will start chapter 8, and we have discussion questions here. So please make sure that you have your copy ready. We also have this set of review questions for 9, 10, 11, right? For 7 again, it will be the quiz 2. So we don't have discussion questions for those. We're good, Ahmad? Please check.
Yeah, it's oh.
Oh, all right, sorry.
Okay.
Any other questions? Remember, the assignments are here in this folder. And I've indicated like what chapters are covered in these particular assignments, as well as the due date. So for us, if we say we've finished chapter 8, since the information is say still fresh in your minds, then you can look at the questions for chapter 8 for the assignment. And yeah, make your notes and start working on it. These are due dates. You can turn this in way before the due date if you're already done with them. All right. Okay. Any other questions? I will not start. I know we have about 30 minutes left. I will not start chapter 8 today because I thought of giving you the rest of the time for the rest of the time for today's class for you to finish up maybe your assignment 2, which is due tomorrow. I hope you can still hear me. It said that my internet connection is unstable. Right. So yeah.
I guess one last question.
No problem. All right.
For the coenzymes, do you want us to memorize their structures as well for the test?
For the test, recognizing the structures will really help Aman. Yeah. And knowing if a coenzyme is a prosthetic group or a co-substrate. And those terms that we covered in chapter 7, those are also a must know. What is a prosthetic group? What is a co-substrate? Etc.
Okay,
So
thank you.
it will be like, all right. So remember, class, the quizzes that you do, those are like samples maybe of how exam questions may be asked. So for your quiz 2 again, like this one here, well, you'll be able, when you do your quiz 2, you'll be able to refer to your notes, right? But on the exam, what if I give you some of the structures of coenzymes? Will you be able to identify which coenzymes that? ATP, easy to remember, right, class? You should be very familiar with the structure of ATP. Is it a co-substrate or a prosthetic group? How about S-adenosylmethionine? If I put the structure of SAM there, will you be able to identify it? What if asked, what is the main role of S-adenosylmethionine? Transfer methyl groups in reactions, right? So yeah, Ahmad, that I hope that helps. I hope that answers your question.
It did.
All
Thank you.
right. Any other questions? So this part here, if you would like, what would you like? We'll end here and then you work on your assignment, or what I can do is get you into your groups if you want to start on this. So you'll kind of, this is a very short one. This is a very short quiz. So there are actually just two main questions here, right? So just let me know. What do you think? Do you want to go to your groups now? And you can also, for your assignment too, it's fine with me if you confer with each other for this, but then you have to do just like your quiz, submit your work individually, right? Yes, any questions, comments?
I think I'm gonna work individually for right now and just talk to my group if I need any help.
All right, okay. All right, so have a good day, everyone. Those who want to leave now, if you have questions, I'm here. So Ahmad, okay, have a good day. See you tomorrow.
Okay, see ya.
All right, thanks.
I'm gonna do the same. Have a good day, everyone.
All right, thanks, Fanag, Ali. Okay, thank you, Anderson. Have a good day.
Have a
Thanks.
good day.
Thanks, Isis. See ya.